Okay, picture this. You just walked out of your Cambridge Biology 9700 exam. You saw the question on comparing prokaryotic and eukaryotic cells, and you immediately thought, yes, topic 1.2, I know this inside out. You listed the differences, you talked about the nucleus, the ribosomes, the cell wall. But then results day rolls around and your grade is, well, entirely different from what you projected. Why? Because most students are losing easy marks on this exact topic. And it's not because they don't know the biology. It's because of one tiny, fatal mistake in their exam execution. So let's dive into this. Because after this explainer, you are absolutely never going to make that mistake again. Seriously, we've all experienced that absolute shock, right? You look at the mark scheme later and realize your seemingly perfect, detailed answer just got heavily penalized. You wrote down literally everything you memorized from the textbook about eukaryotic organelles and prokaryotic structures. You felt confident. You felt like you totally aced it. But here's the thing, Cambridge examiners aren't just looking for a memory dump of facts. That is a massively dangerous trap to fall into. They're testing a specific set of cognitive domains that go way, way beyond simple recall. So, the crucial point we need to get straight is this. Knowing the facts is really only half the battle. The reality of these AS and A-level exams is that they're high stakes. The assessment objectives for Topic 1.2 target very distinct cognitive domains. We're talking knowledge and understanding, yes, but also handling and evaluating information, right alongside experimental skills. If you're just listing facts about, say, peptidoglycan cell walls versus cellulose cell walls without applying them to the actual question prompt, you are throwing away easy marks. It is all about your exam execution. All right, let's get into Section 1, the costly exam mistake. To truly understand where things go wrong, we went ahead and dug straight into the official Cambridge Principal Examiner reports. These reports are gold because they tell us exactly why students failed to reach that top grade boundary. Look at number 1, missing units in cell calculations. When you're comparing prokaryotic and eukaryotic cells, you're constantly dealing with magnification and actual size calculations. Writing down a number without the micrometers or nanometers, boom, instant mark lost. Number 2, failing to check those units and scientific terminology. And number 3, which is honestly the most dangerous, writing answers that ignored the specific context given. If the stimulus gives you a novel strain of bacteria, you cannot just write generic prokaryote facts. You absolutely must anchor your answer to that specific bacteria. And this right here brilliantly illustrates the difference between an average student and an elite student. Do they just memorize? No way. Elite answers do not isolate cell biology in a vacuum. The theoretical architecture of Topic 1.2 definitely rests on precise definitions and causal relationships, but more importantly, it rests on integration with other syllabus areas. So if a question asks about eukaryotic cell structures, a top-band candidate will actively integrate relationships from topics like protein synthesis or cell signaling. They connect the dots. It's like a giant puzzle, and the examiners reward them heavily for putting the pieces together. Moving right along to Section 2, the procedural framework. In just a moment, I'm going to show you a hidden examiner clue that literally changes everything. But to make that clue obvious, you've got to master this core procedural sequence first. Let's break this down step by step for systematically tackling literally any cell problem. Step 1, deconstruct the question. Identify exactly what is being asked. Step 2, extract relevant data from the stimulus materials. Please don't ignore the graphs or tables they give you. Step 3, select your analytical framework. Are you comparing? Are you calculating? Step 4, execute your analysis with clear, logical working. And finally, step 5, present your findings using precise, Cambridge-approved scientific terminology. This systematic processing is exactly how you build a bulletproof answer. Now let's see how this builds into your ultimate safety net, the method mark strategy. The examiner reports explicitly state that students lose marks by simply not showing their working. Think about it. Even if your final calculation of a cell's magnification is completely wrong, maybe your thumb slipped on the calculator under exam panic, it happens, showing a correct analytical process can actually save you. Method marks are awarded for the process, so never ever just write down the final number. Always show the examiner exactly how your brain got there. Okay, on to Section 3, flawed versus perfect answer. Let's apply all that theory directly to a Cambridge-style written response. We're going to look at how a student who just memorizes facts answers a question compared to a student who actually uses a systematic framework. This right here is your 4-tier formula for the ultimate top-band answering strategy. Whenever you're faced with a complex descriptive or analytical question, follow this exact path. First, state the biological concept clearly. Second, pull hard evidence or data directly from the prompt stimulus. Third,
Apply your scientific reasoning to link the concept to the evidence. And fourth, polish it all off by matching the specific examiner wording and terminology required for topic 1.2.
This brilliantly illustrates the transformation. Take a look at the flawed answer. It just lists generic cell differences. Prokaryotes have circular DNA, eukaryotes have linear DNA, without any specific context or units related to the actual data provided. The examiner reads this and gives it what, maybe 1 mark? Now check out the perfect answer. It integrates the concept, pulls evidence straight from the specific table provided, applies reasoning as to why that difference matters for the cell's function, and uses precise terminology. This completely takes a weak, generic answer and elevates it into a maximum mark response. It's a total game changer. Which brings us to Section 4, Statistical Analysis and Evaluation.
As you move into higher-order questions, you aren't just identifying organelles anymore. You're actively handling experimental data, analysis, and evaluation. You need to know exactly which statistical test to pull from your toolkit depending on the biological data sitting right in front of you. If the exam asks you to compare the mean diameter of a sample of prokaryotic cells against a sample of eukaryotic cells, you use a t-test. If you're dealing with categorical data, like the presence or absence of a specific plasmid across different bacterial colonies, you use the chi-squared test. And if you're looking at relationships between variables, you use a correlation test. Selecting the wrong test here will instantly cap your mark band, so you gotta know these.
Beyond just running the numbers, higher-order problem-solving relies on a deconstruct, solve, evaluate model. Evaluating the quality of evidence actually requires critical analysis. You have to actively identify the limitations of the experiment provided, note the assumptions the researchers made, explore alternative interpretations of the data, and critically assess how all these factors affect the final conclusion. This evaluative level, this is exactly where the A-stars are born. And finally, Section 5, the examiner cliffhanger. Okay, let's circle back to that hidden clue I promised you earlier. You can know all the biology, all the formulas, and all the working, but every bit of it falls apart if you misread the very first word of the prompt. Seriously, what actually happens to your grade when you mix up the command words explain and describe? It's a massive diagnostic trap, and it will drop you to 0 marks for that entire section, even if everything you wrote is scientifically factual. You absolutely must understand how command words secretly decide your mark band. We unpack exactly how to decode them in our next explainer, so jump straight into that one right now to ensure your exam strategy is entirely foolproof. So what are you waiting for? Keep learning, keep applying the framework, and I'll see you over there.